Hello!
Hi.
Welcome to Microbiology Twitter Journal Club, where we nerd out big about all things small.
My name is Danny, and in a previous life, I dropped out from a PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Nowadays, I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit based in NYC dedicated to enabling the public with the tools of biotech.
My name is Faz, and in a previous life, I got my PhD in microbiology from Imperial College London, where, among other things, I was making flesh-eating bacteria glow in the dark and doing some work on vaccines. Later, I worked as a research integrity specialist, and these days, I'm working as an editor for an academic journal. And in my spare time, I'm a science comedian, but today we're going to be looking at the facts and we're nerding out, so I'm going to be on my off day when I'm being funny.
So every week we meet to talk about microbiology. Today's our news week, where we go do an overview of papers we've seen in the news or other sources to find an article that we can cover next week in more detail.
And next week will be our deep dive week, where we take one of the articles we analysed today, and then we will dive into it figure by figure to really get into the nitty gritty of the data and learn what it can and cannot tell us, and to figure out how science is done. So make sure to subscribe to satisfy your microbiology curiosities.
So you can follow along with us with the papers that we discuss on either week in our shared Zotero library. It's linked in the doobly-doo below. Um,
Yeah, and we want to hear from you, so please use the comments to chat to us or tweet us using the micro_TWJC hashtag. And yeah, let us know if there's any papers you want us to look at as well.
mhm. Yeah, so first up we have this news about the B.1.1.7 variant.
Yes. So yeah, this is a pretty big news story in the UK, and I think it's so we talked
I think
about it.
I think globally, I've seen
Yeah,
right everyone's talking about it now. Mutation. It's a touchy subject.
yeah. I know our last video on it did really really well,
Mm,
which is both good news and bad news. I feel like in an ideal situation we'd just be like a boring science paper, but now people are actually genuinely interested in it. I'm like I feel guilty about we're making we're getting views off of
well, it's
this.
good. We brought together stuff people can read, right? That's important.
Yeah. Um, So anyway, the first paper I want to bring up is this paper. Well, it's not even a paper. It's more like a preliminary report called
Mhm.
titled "Lineage-specific growth of SARS-CoV-2 B.1.1.7 during the English national lockdown." So this paper charts the spread of the SARS-CoV-2 B.1.1.7 variant between like the 1st of September to the 22nd of December. So it's a long period of time, and during that time, the UK went into its second national lockdown. So basically, Boris was like, "We need to go into lockdown so that we can keep doing Christmas." So, uh,
Yeah, I mean that's the concern, right? Like it's where things intersect with public health, right? You know people are going to be moving around during this time. When I left for the holidays, we heard on the radio that like they did a poll. It's like people were like, despite the lockdown, people still wanted to go and see friends and family.
yeah,
So,
yeah, this is like quite interesting because like the first national lockdown was really strict, really stringent. The second one, there were certain things that were different. So schools and colleges and universities remained open. So there were certain things that you were still allowed to do. And this one kind of charts the effect that had not just on like on the whole SARS-CoV-2, but also on the prevalence of this particular strain. Um,
mm.
so I'm listening
And
to
we're
people.
seeing this is that funny website again that came last Newsweek, right? Like not quite a journal, but it does seem it's presented very much like a journal.
Yeah. So this is where the COG UK, the group that is organizing that's been like monitoring this outbreak from the start. They've been
Mm,
putting all their preliminary stuff on this website, and
yeah.
yeah, so it's kind of an odd sort of place to put it because it doesn't have a
Right.
DOI.
I mean, I think it's a really good thing. It kind of like it's like it makes it easy for these authors to publish and control comments and like see
Yeah.
responses and stuff to their work. Hmm.
Yeah. So we've got a bunch of graphs, and I know that this is. I'm see if I can put this in a bit bigger, but uh, essentially like the big like the green line is the normal SARS-CoV-2 strain, and the red line is the new SARS-CoV-2 strain. And so during the national lockdown, so that in the kind of grey areas, you can see the green line takes a dip under, and what the dashed line is for the R, so that's the reproductive number. So once you get that under one, then you're basically controlling the outbreak. Um,
Hmm.
so the green line seems to be controlled, but the actual red line is not controlled. The red line is still above that R1, so that means that was still spreading during the lockdowns. And
Mm,
they do some like data analysis and basically calculate this new variant has a higher R value, and that
mm,
this increased transmissibility made the lockdown less effective,
sure.
because this was just so much, and yeah,
Yeah,
so
because I bet that when the model of the lockdown was like initially proposed, they probably didn't have, right? They made assumptions about
yeah,
certain things, and like maybe those are a little bit different. Like it seems like we've been hearing stuff that the properties of this variant may be slightly different, right, than classic SARS-CoV-2.
yeah,
It's like, so I can see that this one would spread over the other one.
yeah. So this has got fairly sparse genomic data, so there's a lot of modeling and mathematical models just trying uh, to pick out what's going on. But they do
Mhm.
provide like some evidence for increased infectivity of the strain, and kind of to justify why the current lockdown. So we're now in the third lockdown, and it's being much more severe than the previous ones. Um,
Mhm.
but that's
Yeah,
so yeah,
it's good. It never hurts to have more compliance with public health, right, recommendations. And if this is the information that can convince people about that, then that's probably a good thing. I will say that this information looks really similar to when we were seeing I think I said this last week too, the D614G, right? Like we were seeing the very similar types of graphs, right? Where you see the prevalence of a variant
yeah.
kind of take over, right? The existing ones.
I
Uh,
feel like D614G didn't have such a drastic change in the R0. I think most of them would seem to be like competing with other strains so much. So
yeah,
it wouldn't so with those it wouldn't be so much that the lines are different. The lines would be like almost add up to the same thing, but the way they'd wave. Whereas like with these two lines, if you kind of add them up, they'd be bigger.
yeah.
Whereas with
Sorry, I don't mean the R0 graph. I mean the top graph, the one
oh
that has just like new cases,
yeah
right? Like those in that new cases graph, right? You can see it's between the red and the green,
mm,
right? So like the red variant is above now the other lineages,
yeah
right? Indicating that it is the more prevalent version, right? Like
yeah
something's happened in the last it's pretty small window of time, right? In the last two
yeah
weeks or something, at the beginning of December, where now this is gaining prominence. So that would be the indication. That's probably the types of differences people are looking at when they're making this call and saying it's more effective, it's more
yeah
transmissible. Mhm.
yeah. And I think that's going to bring us to the second paper, which is from an external group that also did some work trying to estimate the transmissibility and severity of the SARS-CoV, estimated transmissibility and severity of novel SARS-CoV-2 variant of concern
Yep.
2012 numbers 2020-12/01 in England. So this is again, it's quite clearly not peer reviewed. It's still, but they so this is while the previous paper came directly from the COG group, this it was very preliminary. Well, this one is like a bit more filled with stuff. So they got like they're looking at the same data, but they also combine that with mobile phone data to estimate the
Mhm.
number of contacts, and then put that into the modeling to see how effective that. Because there's always that theory that maybe it wasn't the strain's more transmissible, maybe it's that the lockdown failed. Um,
Yes.
so this the aim of this is almost to try and
Yeah,
like
to really,
analyze
I
that.
mean, yeah, because again, we only have this one window, right? But before we can do any animal testing, we only have the window of this epidemiological surveillance that we're up to.
Yeah,
And so, of course, you got to do the right controls because that data is not is not gathered for the purposes of determining this information, right? You have to remove other possibilities.
yeah, and I think that this was published on. Let me double check this. Yeah, this was published on December 26th. So to put that into context, this strain was announced on December the 19th. So
Mhm.
this is equating to about a week's worth of like work that
Yeah.
had to be put in very rushed. An animal study requires like depending on what you're doing, you need to order animals, you need to breed them, you need to make sure. So animal studies might take
Absolutely.
about a
Yeah,
month or two.
won't have those yeah, and we said this last news week as well, right? Like for people who make decisions, right, in policy and so forth, right? You kind of need faster, you have to make your decisions on like limited amounts of information. This is the challenge, right, of navigating our society using science, and like it's a good way, right? Because we can verify and we can like go back and say what did we do right and wrong based on how we acted. But we do have to sort of just take the little hints that we get and then make a plan from that.
Yeah, I mean, this actually does a lot of that planning, and they don't just monitor the outbreak so far. They also do make some predictions. So they
Mm,
predict, they try to analyze like what kind of um, what kind of strategies to managing the outbreak we should take in future, and kind of predict how ICUs will be overwhelmed based on this new strain and this new R number.
sure.
Because um,
Yep.
while this variant
Yeah,
isn't,
updating the models with this information
yeah,
and then yeah, regenerating yeah, a set of best practices guidelines.
yeah, because I mean, people will be getting it faster, so ICUs will be more likely to be overwhelmed faster. Also, the herd immunity threshold may have to be much higher. So you have to put that into calculations of how many people need to be vaccinated, how many people need to be put in isolation. So this is quite
Yeah,
an interesting paper from that decision-making perspective of like putting yourself in the
sure.
position of the policymakers, looking at this, and the information isn't perfect, but you still have to make a decision on it, and that's a lot
Yeah.
of
Yes. That's the scenario we've been navigating probably for this entire pandemic.
yeah. I mean, waiting and seeing often is until all the data's in can create a massive delay that means all your things you can do are kind of less effective.
Yes, exactly. The boat has sailed on those things, right? Like
Yeah.
we're okay, well, up next we have
S
S
gene.
gene dropout patterns in SARS-CoV-2 tests suggest spread of the H69 deletion V70 deletion mutation in the US. So
Yep.
this is like early surveillance in the US now of the same one of the loci that is involved with the B.1.1.7 variant.
Yeah, I found this interesting. It comes from a genomics testing company. It comes from Helix, and so they've been doing
Hmm.
some of the COVID testing in the US. And
Mm-hmm.
and one thing that is known about this new strain is that the H69 V70 deletion can stop the kind of primers that bind to that gene from working. So the specific test for the S protein stops working sometimes if you've got that deletion.
Gotcha.
So what they've been doing is they've been looking at other genes as well. So throughout the outbreak, they haven't just been looking at the S gene. They've also been looking at the N and or ORF1AB genes. So they've
Hmm.
been, and so based on that, so since like this outbreak is going on, they've been doing this like test for three like different genes, and they've noticed that there is an increase in the dropouts of.
Yeah, for this one this one loci that they test. Yeah, I mean this is sort of a great callback to our discussion from last week, right? When we were talking about coverage, right, over the genome and testing. Yeah, very exciting stuff, like in terms of just letting people know that, not all that you're gonna have to cover more bases as the virus changes, right, for these diagnostics.
Yeah, the interesting thing is like so when they heard about this new strain, they went back over their records and to see the number of tests that register this kind of dropout, and they found there was like a
Hmm.
spike in October, which kind of
Hmm.
rose to in November in various states. And
Hmm.
well, they do kind of clarify that this doesn't necessarily mean that new strain is in the US because obviously this deletion mutant has evolved multiple times independently.
Yes, that's
But
right.
so it could be a new strain, but the timing is the key because that peak in November also kind of correlates to the when the B.1.1.7 strain arose. So it does seem
Hmm.
to be like kind of more than a little coincidental that these both appear at the same time. So uh,
Yeah.
it
I
could.
mean, I would say this, right? Like, right, mutation and selecting for this terrible experiment of like of human people, right? Selecting for those mutations, it requires like a lot of people to have infection, right? So if your country has a really high amount of infected individuals, there's more of a chance, right, of seeing mutations. And if those mutations are beneficial, as we're seeing some evidence that like at least when put together, they're beneficial in the B.1.1.7 variant, then they'll spread in the population, right? And
Yeah,
then maybe they'll until you kind of settle on the successful variant, and that one takes over.
yeah. Um, so yeah, I mean they've got this map of the US on as Figure 2 where they show like the proportions of times that strain appears, and
Mhm.
so there are peaks in certain areas, and the one thing I'd like want to know is like whether how systematic the coverage of testing is because there might be some areas where they don't do much testing at all, so they wouldn't know. Um,
Yeah.
So, I mean, the US is a giant country with lots of different like states that have their own systems. So, as an outsider, I find it very difficult to navigate this sort of thing. Um,
Yeah, I mean, the other thing that I'll say is complicated about this is just from their company's data, right?
yeah, exactly.
So it's like, so hotspots also appear proportional to how much they're collecting in those areas.
Yeah.
Yeah, because there's like a patchwork of companies that would do testing probably.
Mm, yeah, I mean, because one company isn't necessarily big enough to do the entire US. Uh,
Which again, this I mean, I don't want to throw so much shade on the American healthcare system, but like that is a weakness, right, of non-public healthcare systems, is that it sometimes it may be harder to unless there are open standards that allow
mm.
that transmission of that information. It may be harder for someone to do the single analysis and gain like a trend across the entire
I
country.
mean, it's also like a massive country just in terms of numbers. So there's a
Oh,
lot of
absolutely,
infrastructure that needs to be put in.
absolutely,
So I think a lot of stuff does need to be handled statewide. Um,
yeah, yeah,
but so like I'm going to be flipping off to another papers because
yep.
the
Let's.
UK has a similar sort of system. So this is from a UK test and trace lab that has also been monitoring dropouts. Um,
Mhm,
so like the UK, much smaller country, so again we don't have to deal with the. So this is very much a nationalised sort of event. So it's part of Public Health in England.
mhm,
So it comes from a government-related laboratory, and it investigates the prevalence of gene dropout. But they ask a slightly different question. They look at this thing called the cycle threshold value for RT-PCR.
mhm,
So that's basically the time it takes for. So we talked about this a little bit last week. The time it takes for the PCR test to reach saturation. So
yes,
if
yes.
if you've got
And so,
more,
like, the sooner it happens, the more sample we started with, probably, right? Meaning a
right,
higher amount. This is the quantitative. This is how RT-PCR can be quantitative, right? Because the faster something amplifies to that threshold, right, it means that there was more of it to begin with.
yeah. So they measure the other two genes and see how. So they did the similar kind of tests, but they measure like how quickly those other two genes meet the cycle threshold, and what they
Mm.
find is that in general dropouts tend to reach that threshold much faster. So meaning that they've got more genomes.
Yeah.
So
Yeah. So this is nice. I think this is a nice complementary one also to this idea that this could be a more successful variant, right?
yeah, um,
Yeah, I guess sample collection is like one of the things that I would wonder about this one. Like because it could also be something like what are the criteria in which they admitted samples into this? If it's totally unknown, right,
yeah,
then you don't know. More people are getting. Maybe it makes something about this infection makes people want to get tested sooner. It's probably Sentry though, right? This is just um, like an overview, like a sampling of everybody in the country.
yeah. So this is I think
Hmm.
a sampling of people. I
Yeah.
mean that and yeah, I mean it's not like they're dealing with equal groups, so it's not necessary. And the actual like kind of difference is relatively subtle.
Hmm.
So it's so
Hmm.
wouldn't say it,
It's
and
also, yeah, it's from the same. I also think it's like fundamentally the same data, or no, it's a different analysis, right? Like
mm,
they're trying to correlate. Yeah, the CT is a different readout of success, we'll
mm,
say, right? Success of the virus. So that's good. I do think that this adds like a little minor, right, bump to the idea. And again, taken all together, this is what people are making decisions off of, right? So,
yeah, and I mean
yeah.
it could add another piece of the puzzle of why this strain is more transmissible. Uh,
Hmm.
I mean, I guess I probably should disclose that number of genomes is viral load, so that doesn't necessarily translate 100% to viral titer, but it's still very much an indicative sort of thing to think about.
Yeah.
Uh, and the next is quite an interesting paper. It comes from Pfizer.
Hmm.
So, neutralization of N501Y mutant SARS-CoV-2 by BNT162b2 vaccine-elicited sera. So, will this strain be neutralized by vaccine sera? So
Uh-huh.
if you're immunized with the BioNTech vaccine, are you going to be safe? Um, so they
Of
answer
course,
this.
this one out because
Yeah,
they want to make sure people are excited to use their vaccine.
yeah. I'm surprised none of the other vaccine companies aren't just rushing out stuff because this is like a 3 page long paper. It's very short, but they just like pull out the kind of they've got this PRNT50 data where they pull out and they kind of say that okay, there's not much difference between the old Y N501 value and the Y501 value.
Yeah.
Um,
I think it's good. I mean, this is the type of stuff. Yeah, I agree. Like, to allay fears people for different vaccines, it would be nice to see. But I also think seeing it for one vaccine also sort of makes you think that it would be okay in the other
hmm,
vaccine scenarios.
yeah,
Um, yeah, just because like you're getting the same spike protein mostly in each one, right?
hmm,
And like the idea of this mutation is this one sits in the receptor binding domain. That's like the, oh man, like that zone could be one of the zones that like
yeah,
doesn't recognize. But yeah, it seems I guess this still works for the neutralization. It still neutralizes, right? That's what it's saying.
yeah.
Yeah,
It
still.
still basically neutralizes. I mean, for their sample size, they didn't really see much of a difference in the variation.
Mhm.
So, um,
But again, I don't think that's the primary. I don't think when I heard this news about the variant, that wasn't the primary concern in my mind
hmm,
about the variant, right? Like, oh yeah, I believe the vaccines will all work against it actually. Like just from like my intuition about the science, right? Of course, it's good to see that data like this, but
hmm,
my concern is more that transmissibility or like that it goes to higher titers in people, right?
yeah,
Quicker. Because that just means that it's more difficult to control, right, on the big scale, right? Because I do think that that's the biggest problem, the big problems we've been having. I mean, even the variant itself is a product, right, of the fact that so many people are infected, right? If there were fewer people, we wouldn't see the emergence of variants like this, right? Because there wouldn't be that large population to play evolution in. Um, so yeah.
yeah. I mean, one last thing. They sorry. They look at just the N501Y mutation, but the
Mhm.
new mutant has a bunch of other mutations in it that could. So the H69B70 deletion, like so, last time we talked about it, we had this paper where the N501 mutation like kind of was theorized to increasing by about 20%, increasing transmissibility, and then when the H69B70 came in, that was a much bigger increase.
Yeah, another jump.
So I'd be very interested to see what that effect would have on antibody responses, or just trying to figure out what the actual fuck is happening with the spike protein when that happens.
Yeah, for sure. What's the I mean, like, yeah, in some ways, like the golden piece of information that could help us answer some of those things is like, yeah, this modified spike, because it's all these modifications to spike protein, right? What does it, how does that change its behavior in its interactions that are biologically relevant, right?
Mm.
With receptor. Yeah. And again, those are stuff that probably the experiments are being done now, right? Like
Yeah,
that's the nature of science. Like we're not the first people to think about these things, right? We're talking about them, but like inside of labs, people are thinking about these things, setting up those experiments, but it takes time.
yeah,
And so you have early decisions to try to get ahead of potential problems, and then yeah, and then we have to wait to see some of those confirmations. Well, and nerd out about them and say
hmm,
like what we think is like happening, right? Like try to guess, yeah, exactly what's going on at the molecular level.
yeah.
In this list, you also had structure of SARS-CoV-2 ORF8, a
Oh
rapidly evolving
yes,
immune evasion
yeah. So
protein.
ORF8 is a protein that was again also seen in this mutant. So I looked into the paper. The paper itself is very structure, so it didn't really say anything about this current strain. So I moved it to later in the show. So kids, tune
Gotcha.
in if you're interested in ORF8. We'll have that coming up right at the end of the show. So
Sounds good.
yeah, the curiosity
All right, so
gap.
that's our B.1.1.7 coverage, and so we'll move on to some other news that we've seen.
Yeah, so I guess it's somewhat related, but escape from neutralizing antibodies by SARS-CoV-2 spike protein variants. So uh,
Hmm.
we're kind of worried about escape last time because the B.1.1.7 escape. Maybe not, but is it
Yeah.
possible?
Yeah, and I mean this is very similar. This is like also a callback to our decoys video. Like
Mm,
I feel like we talk a lot about escape mutants in the decoy video because the whole point of making those ACE2 decoys are to circumvent the possibility of escape.
okay.
So yeah, so we did look at a little background here. This is very similar, right? They try to evolve samples of virus in the presence of monoclonal antibodies. So I think that's
Yeah.
important to remember, right? These are antibodies that target one part, one epitope,
Yeah.
and they show that they can occur.
I mean, I think the thing that we also need to be clear on is that they use a pseudovirus, so not real SARS-CoV-2. Because if they did, I
Mhm.
would go mental. Because having
Yeah,
that kind of
that gain of function experiment,
yeah,
right?
I saw it and I was like, no, they didn't, they bloody didn't. Because I was freaking out when they did this in mice. So I mean, it was like, so yeah,
Yeah,
no,
previously, I think yeah, it's good thing to point out. Yeah, previously they did this in um they did it in yeast.
yeah,
When we were looking at the decoys, they did those libraries in yeast, and then they used those as like binding assays and neutralization libraries on yeast bodies, I
yeah,
guess. Yeah, so here they're using a pseudovirus. So I guess that is also helpful, a little bit closer to the size of a virus
yeah,
that we're seeing, but not with the payload that would
yeah.
that would indicate bad experiment to do without really. I mean, I'd maybe never do those experiments, right?
Yeah.
Um,
I mean, so
you have to
it's
be very careful about what you're learning, right?
yeah,
There's a space for it, but you have to know what exactly you're learning when you employ that.
yeah. So I mean, they use VSV and they exchange its normal like kind of entry thing with the spike protein, which
Hmm.
again is quite interesting. The thing is, like, the question is like the mutation rates are much higher in there than in normal SARS-CoV-2 because SARS-CoV-2 has all
Hmm.
this proofreading gear. But
Yep.
so I guess you're right. What you are basically going to do is like see what specifically these monoclonal antibodies. So these monoclonal antibodies aren't just any. They did like kind of take them from a paper which was trying to find a consensus immune response from people and then pick
Mm.
out monoclonal antibodies from that. So they are can
Hmm.
be you can argue that they're important, but I think that this also might have benefited from a bit more sequence gazing. So like seeing whether the mutation they found turned up in the GISAID sequence like libraries that are being collected.
Sure.
Um,
Absolutely. Yeah, that would be nice. That would like relate these findings, right, to something relevant in our right?
oh,
Because right now it's just a model of mutations appearing, but if we could see those mutations in the population, then we could also just like say something about, yeah, do they gain prevalence? Are they fixed, right? Are they ones that we already have?
I mean, just
Yeah.
thinking like from a perspective like clickbaity, like oh the variants that can avoid can be immune to your antibody response are already out there. That kind of thing. It's got that
Yeah.
fear response that that people are looking for when they want to promote like news stories.
But actually, like, it's an interesting, like, I mean, I don't know, it's a complicated relationship, right, one might have with the news and clickbait,
Yeah.
but like, but I mean, that message is not a bad, in my mind, it's like, we have to understand that there is, like, the more the virus spreads, there is more possibility of finding variants, right? And like, the world is doing their best to control that spread and then also surveilling all these genomes, right? And then trying to assign significance to the genomes to make sure that we aren't bringing ourselves to a situation where we're just letting things go.
Yeah.
Um, yeah. So there's some truth in that type of clickbaity title.
Well, I mean, I think like raising awareness about the possibility of something like this happening, I think is kind of important, so that people understand the importance of doing testing and tracing and doing that kind of
Yeah.
isolation. So, yeah.
Yeah, absolutely. Absolutely.
Uh, so the next
Oh,
paper I have up the list is efficacy and safety of the mRNA 1273 SARS-CoV-2 vaccine.
this is Moderna's paper, right?
Right.
I feel like it was like lost in the mix of all the things that have come out. But yeah, they finally published their phase three information. And well, I didn't dive into it at all. I just figured, oh, it probably works.
Yeah, that's
It
basically.
can be approved by the regulatory bodies, so we're gonna see more stuff about working.
Yeah, I mean, I did a brief look over it. Like 94.1% efficacy, they calculate it. They look in older populations as well, but there's a lot of variability in older populations compared to normal. So it kind of mirrors what we've seen with the other vaccines.
Hmm. Hmm.
Well, I mean, with the bio.
This is the
Sorry.
vaccine that my grandmother has received.
Oh wow.
Oh, wow. Yeah, I was like, well, that's really fast because this was not the first one to appear in Canada, but it's the one that she ended up receiving. I guess that speaks to supply chain things, right? Where it's like this one's manufactured in the US, probably.
Yeah.
I think.
Yeah.
Yeah, and so like we're close to the US, so like that's the one that we would get first.
Okay. Oh, well, that's interesting. Very personal. Sorry about that. I hope everything's
It's
going to be
fine.
okay with your grandmother. I'm
It's
trying.
interesting, like what's out there.
It's like, how do I not be a
Um,
cold robot? But yeah. Uh,
uh, let's see. Yeah, I guess what's interesting about it is doesn't seem particularly different from anything else that we've seen.
mm,
Just like rolling through these figures. Oh, they're also doing that thing where they they look at just the first dose response,
mm.
right? This was similar to what we saw in
Yeah.
the previous ones, where it's like, oh yeah, like if you get one dose, it's a little bit there's some effect, but really it's being tested with the two doses. That's where you see the most.
Yeah, you've got about 20 days where it's got without the dose, and there is a difference, but it's quite hard to figure out. Like, and I think we'll talk a
Yeah.
bit more about this later. But
hmm
yeah, I think it's important. That's important data still. Um,
Okay, then we have this one, phase one safety and immunogenicity trial of an inactivated SARS-CoV-2 vaccine. This one is from it's like because I found it because of the news article saying that India approved the AstraZeneca
mm,
vaccine as well as they have their own
yeah,
vaccine. Right? And the only published information they had about it was this phase one, but they also have published the pre-registration for their phase three trial, so that is in
mm,
progress. And this is an inactivated vaccine that's coming out from an Indian company and manufacturer. So that's like the CanSino, right? This is the classic way of making vaccines. Get a bunch
yeah.
of virus, kill it.
Yeah, they don't actually mention how they inactivate the vaccine, so and there are lots of different ways. So I don't know how. But they also add like adjuvants as well, like alum and IMDG. So IMDG is a TLR like 8 and 9 activator maybe. That's meant to activate the antiviral response. So I mean, they do some basic safety like antibody titers, and they took out
hmm
some like T cells, B cells, and just measured whether they produce interferon where when stimulated by SARS-CoV-2 peptides. So yeah, it made the immune cells angry, so that's good. Like the yeah, when when cells
it successfully pokes the wasp's nests of our immunity.
yeah, so yeah, I think that's
um,
yeah.
Yeah, I mean, this is we haven't really talked about this these inactivated vaccines. There's been such a rush of like these new vaccine platforms that like there's not a lot of focus on these ones. But it's interest. I actually find it interesting that they use that they put so much focus on the adjuvants, right? Because I mean that is the sort of classic frontier of vaccine R&D,
Mm,
right? If you're making inactivated things, is playing with those adjuvants to try to get the best wasp nest poke that you can muster.
yeah, I feel like I have been trying to do some more about the inactivated vaccines because they're like the classic vaccinology, but there just
Yeah,
isn't much data out there about them, which is
right.
crazy.
Yeah.
Because
hmm
like in theory they're the ones that should be the quickest to develop and quick and they're being rolled out, but I just
Right.
haven't been seeing that data. And I mean, honestly, they've got the potential to be to be really good. So I just want so it's just
Yeah.
baffling that
Also very cheap to make and
yeah,
manufacture, right? You're
yeah,
growing a bunch, you're inactivating it.
yeah, and you can store it in refrigerator temperatures. I mean, there's a lot of good things about them, but we just don't have any
Yeah.
data about it, and that's kind of frustrating for me because obviously
Sure.
like those are really
I just
important.
right now as we were chatting, I'm like, oh, we're talking to the audience of the internet. Like, also, I think people should realize that making this vaccine is not a dangerous thing. Growing up the virus, right? They don't have to grow up a huge amount of volume. They could do a continuous reaction where
Mm.
it's a very small amount of volume, right, of the virus, and it's being inactivated as it flows to the next thing. It's not like they have giant vats of active virus around that could like spill out or something like that.
No, I don't. Yeah, that's because firstly that'd be really expensive to do as well. The much cheaper to maintain a few small cells at a low volume and then just keep inactivating them. Uh,
Yeah, exactly. Okay, let's see what we got. We have prior SARS-CoV-2 infection is associated with protection against symptomatic reinfection.
so yeah, this is a letter to the editor, right? And it's it's quite important because they look at like healthcare workers who
hmm
tested positive early on in the outbreak and then were later like tested again, and they found like kind of 0 out of like 1,000 people with positive results compared to like the control
Results
with
for symptomatic, right? They
for
could
symptomatic.
still
Yeah,
be PCR positive, right? Asymptomatic,
yeah, that's true.
meaning that they could be asymptomatically spreading it, but yeah, they didn't have symptomatic, which is what we expect, right? Um,
Yeah,
this is the power of our immune systems, right? Is that like if you've if you've survived infection once, you probably will survive it again.
yeah. I think this is kind of important because we have caught lots of like individual like reinfection studies, but actually trying to
Mhm.
put that into perspective, like how common is that to happen? And if you've had like SARS-CoV-2, are you gonna be safe to go out into the public? Are you going to be a carrier still? I mean, this doesn't necessarily answer the carrier question, but it does answer the safety question, I feel.
That's right. Yes. Yeah. It doesn't answer. Yeah. Basically, yeah. If you have had a positive case, it's unlikely that you're going to get sick again
Mm.
in a serious way. It's still unknown whether you will get other people sick based on this information. And actually, this is where I hope that the and again, maybe it's like this is reading too much into the data, but like I was just thinking about the asymptomatic data that's being collected in the AstraZeneca thing. Yeah,
Mm.
I'm kind of waiting the final publication of that
Yeah.
paper, but like I remember seeing that like the it didn't impact as much the asymptomatic rate, right? Like there was a difference, right? Like you could there was more people that got it asymptomatically. It prevented more of the symptomatic disease. And I just wonder what drives that. And is that something that we can, is that knowledge that we can apply into these scenarios to try to answer this everyday question, right? Like once positive and symptomatic and recovered, right? What does that say about your asymptomatic potential?
Yeah.
Hey,
Right.
that's what's always on my mind these days,
Yeah.
right? Because it's like that's like the one of the biggest features, right, of this virus and the way that it spreads, and like that's caused so much consternation for everybody involved.
Oh.
All right, let's see what we got
Yeah,
next.
alternative dose allocation strategies to increase the benefits from constrained COVID-19 vaccine supply. So
Hmm.
this is a big question in the UK because they recently have been discussing giving people just a single dose and then delaying the second dose just to make sure that as many people can get their first dose as possible. And then
Hmm.
and this is a modeling paper that kind of predicts how many more lives could be saved if we did that strategy instead of um, just straight sticking to the standard uh,
Hmm.
one dose and then second dose, and we don't try to expand the vaccine supply. So it's interesting. Uh,
Yeah, I
so
think it's I mean, again, it's one of those things that helps inform policy decisions, right?
mm,
And once again, this is stuff that the companies have made explicit in their publications,
mm,
right? That there is some sort of effect from a single dose, and that is why this paper exists, to help people make decisions based on
yeah.
all the information there.
So they make a few assumptions here. They assume that like say after about I think after 6 weeks they assume that your immunity will go back down to baseline, which I think is kind of very conservative. I don't think that I think that's a very like kind of negative view of what the vaccines will be doing. I think they might be a bit better than that. And but
Yeah,
that
6 weeks is very short. That's like you only get a very small window.
yeah, and they also like assume that if you only get one dose, you only get 50% of the immunity. And I'm not sure we have all the data yet to support that, or whether. And I know that like I think the vaccine companies are saying that
Oh,
you might.
they make an assumption on that. They don't pull that from the actual
I think
data.
they pull that from some data, but because if I saw like a comment from like someone saying that that the vaccine companies are stating that it's a bit more than that. They might think that they're saying that the first dose can produce up to 80% of the protection.
Hmm.
Again,
Hmm.
I don't know whether the data is on that, on that. But I think the
Right.
important point is that this paper is being very conservative, and even then it says that you can avert 23% more deaths by being flexible with this strategy. Um,
Hmm.
so that's an
Yeah,
interesting,
I mean
yeah.
I think it's a really good point. And again, speaking to this point of distribution, right, where it's like it's clear that there are many issues with
Mm,
distribution. So yeah, things that can put public health benefit, locate it right in that process is good, right? Because that's the process that might need help at this point.
yeah. But I mean, I should point out there is like other sides to this argument as well. There are concerns that that these like low dose, having only the first dose means that you don't get the booster, so your immune response isn't up to full like snuff. I mean, the antibody
Mhm.
titers aren't going to be as high as they could be, and there are
Yep.
concerns that could lead to more resistant virus appearing. So
Sure.
again, like having like I think we talked about it a little bit, like with people with weakened immune systems having more likely to have a virus evade that can evade the immune system develop. Because you've got that
Hmm.
half immunity that means that the virus is still able to survive underneath the threshold, and then based on that kind of selection pressure, they can develop resistance. So uh,
Sure.
that's like one of the main concerns
But
about this I've read about.
but I mean, I think that it's kind of crazy, but this is where I think it really helps to have some clarity. What is the vaccine for?
Yeah.
Right? Like what specifically are we using the vaccine for, right? In our world? Is it like because I think there's a tendency to treat it like we did the Swiss cheese model, right, at the end of
Mm.
our vaccine talk because there's a tendency to treat it as if it's like, okay, vaccine, end of story, right? Like once
Right.
everyone's vaccinated, the story is over. But it's really just a very specific strategy. And like
Mm.
the way I think about it is actually vaccine is preventing the most vulnerable people from getting severe disease. Right?
Yeah.
And then like that's all we got from the vaccine. And then everything else is still through mask mitigation and stuff like that. And even when we have our masks, even when we have our vaccine, we're healthy people going out there in the world, we're still wearing our masks until we get the green light that we know that, right, like a huge amount of people have been vaccinated and so forth. So yeah.
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 This is not I guess maybe the point of that is just to say um like these are very policy, like these pieces of information are very much geared towards making policy, right,
Mm.
decisions, and like how it affects our day-to-day decisions, not as much, right, because like we sort of have to see like on a big scale what the strategy of using the vaccine is. um mhm.
Mm. Yeah. I mean, I flip on this like a fair bit of because
In your
sometimes I
mind,
think
yeah.
in my mind I flip flop like a freshly caught tuna. I'm just like sometimes I'm like thinking, okay, well we need to think about saving people as much as possible, but and then another thing is like, what if some people never get that second dose, and I kind of like
Mhm.
there's all sorts of like complications that can happen, and it is a gamble. You have to make this isn't perfect information, and if you're in charge, then you have to just make a decision on it, and you either have to take
Right.
that because you either have to accept the kind of the that you have a set number of people you can save, and then accept that, or you can try to take a gamble on this new strategy, and
Yes. Yes.
so
But I guess and importantly, but if the rate is low of infection, right,
mm,
then you don't have to make those difficult calls, right?
yeah,
Because like this strategy becomes more attractive as, right, rates are increasing and we're like, oh my gosh, like all these new cases, how are we going to deal with them all? But if the cases always remain low, right, at a certain threshold, right, and I don't know what that is, but like if the cases remain low the threshold, then you never have to make that decision, right? You can just deploy the vaccine as you should because you don't have to worry. People are paying attention and like not spreading it around.
yeah.
Um, so
If yeah, if only we lived in that world, Danny. I mean,
right, I mean that's sort of what I'm saying about this information. Like it's like every piece of information is really just like, okay, but I should be the most conservative I can. Of course, like financial concerns, right, are like the primary reason in which that you would not be conservative.
mm,
So health reasons, right, are a big reason where you wouldn't be so conservative. But like if you can err on that side of being conservative in the way that you behave in the world these days, you contribute to keeping those cases low and thus alleviate some of the difficult decisions that policymakers might have to make down the line,
mm,
right? That have these trade-offs in really like people's lives in some ways.
yeah,
I mean, real ways actually. Healthcare is always about people's lives.
mm,
Okay, okay.
yeah. That
Uh, that is all we have for vaccines. So into testing.
yeah,
Uh, last week we ended up talking about, oh yeah, like this testing technique, right? And I remember talking about, oh, but they still have to purify the RNA beforehand, and I heard there were shortages of purification columns and those types of reagents, so I found this paper to sort of see what was the frontier in that space. A fast and accessible method for the isolation of RNA, DNA, and protein to facilitate the detection of SARS-CoV-2. Basically, they have a buffer that they made.
Yeah, apparently they've got a different mix of chemicals that are readily available, and
Uh-huh.
so that are not there's not a global shortage of them, which is good.
Yep. Yep. And so you
And
can mix them up, do your lysis step in them, and then precipitate out the RNA from that mix.
yeah, and I think it's quite interesting because this group was also developing CRISPR test, and they developed this just to help them with that. So I think it's kind of interesting that they're already thinking of that sort of thing, and it's a nice kind of companion if you've read like last week's paper and are wondering, okay, how can I want to apply this in real time? I've
Yep.
I've bought my phone, I've bought a thing. I want to do my own home CRISPR test. How can I
Yep.
how can I start testing COVID? I mean, this is a great companion paper to that.
Totally. Totally. Yeah. Bridging the gap. Here's a complementary method that can be employed there.
Yeah. Um. So
Okay. Optimal COVID-19 quarantine and testing strategies. Um, yeah, so another one that I ferreted out, and I guess this is what puts me in the mood, right, for thinking about like these public health things. Again, this is just a model
mhm.
that they deploy on some data, right? I believe it's a data set that they're fitting into this model, and just trying to see like for if you're tested before you enter the country, before the entry moment or exiting quarantine, before you enter quarantine or after you exit quarantine, where can you employ a test to shorten that quarantine period, right? Because 14 days is this like standard
Yeah.
quarantine period. That's the conservative estimate of the full course of infection. But people want to maybe use testing to try to
Yeah.
dis and people have, right? Like that's why they could do this study because there have been policies that say, oh, test to get out of quarantine or test before to yeah, to lower the amount of time you have to stay in.
I think that's a kind of an interesting idea because I mean, so if I say, so if I'm going to like say New Zealand or I guess Canada, you're gonna make me quarantine for 14 days, or in the UK, I guess if I'm exposed to someone who has SARS-CoV-2, they're gonna make me quarantine for 14 days, and I guess adding like having a test as I go into quarantine could maybe save time if it goes like, oh, you're negative, but again, there's always
Yep.
this concern that okay, it could be a false negative, and
Yep.
also, like testing people when they come out of quarantine is actually kind of a no-brainer because in that 14
Yes.
days, like not everyone's going to have the same course of infection. So making sure that you don't have the virus when you're about to walk into society and go hello, you need to make sure that you know you don't have the virus.
Yep. Yeah, and it's one of those things again. It's like these are things that sort of impact like resource management, right? Because like testing is a finite resource at this moment, at least, and so like finding the best strategy that both protects right people but also allows people the most mobility, I think, is what we're trying to get at here. I've experienced two strategies, right? Like I've done the full 14-day quarantine when I've traveled, and then I'm now doing this one where it's like you wait a few days and then you get tested.
Yeah, I think they kind of hedged towards a being tested strategy that if you like wait for 7 days and then you've got if you got the infection, that's enough time for it to start showing up, and
Yeah.
then you can get that test and confirm
Hmm.
it, and then test as
Hmm.
you exit. Or and if you don't, then you can shorten your test and you'd be like free to go out into the world. Um,
Yeah. Yeah. And
so yeah,
I mean, this doesn't even have to apply to traveling between countries, right? Like, if we're being really careful about this control of the pandemic, we could apply it to traveling between counties or like
yeah,
provinces. What do they call regions in the UK, actually?
counties we call them.
Counties.
Yeah, so yeah, I think that's an important like thing to think
Yeah,
about.
this type of information applies to all these different levels of organization, right? And like integrating that across like larger and larger spaces is sort of the key to controlling a global event.
Yeah, yeah. So
All
next
right.
paper.
Oh, SARS-CoV-2 transmission from people without COVID-19 symptoms.
Yeah.
So yeah, we know this can happen. And this is as I was saying, like sort of like what's caused so much consternation with the control of this virus is because um, it wasn't fully known at the beginning. Like, I mean, I think there were indications at the very beginning of the
Mm,
thing, but it certainly wasn't common knowledge.
mm.
Hopefully, by this time, it is common knowledge. But just in case it isn't, here's a paper that gives us more data to show us that this is the case.
Yeah, this is a modeling paper that's kind of based on a meta-analysis of studies on the incubation time of SARS-CoV-2, and they use that to build a model to kind of demonstrate, I think, about half of cases are related to asymptomatic transmission.
Mm-hmm.
So, I'm
Yeah, because I mean, you can't directly, this is something that you
mm,
can't directly, like, this is the frontier of answering this question because you don't really directly observe this question ever.
yeah, like it's hard. Yeah, people. It's hard to like take that test of asymptomatic transmission with people because again, COVID-19 testing reagents. There's a shortage. We don't
Yeah,
necessarily have the time to test every single
yeah,
person in the country. So
nobody knows yeah, like, you don't know where you got the infection from. And then even if you did have all those contacts, those people have not been tested.
yeah,
So like, yeah, basically it all is very much like high level inference has to be used in order to get at this question. Um, and yeah, so you have that model that gives us this information.
yeah.
Yeah, it's because it's a model paper, I am very ill equipped
Oh yeah.
to
I
give an overview on how exactly it shows us that. If we choose it to read, then I could say,
I
but
mean, I'm gonna say I'm not really I'm not too happy about model papers because any model paper that doesn't have validation on is like kind of very it it's because with mathematics you can do a lot of different things. There's a lot of freedom to like use
yeah,
test whatever assumptions, but trying to make sure that those assumptions relate to the real world is a really kind of it's not an easy thing because sometimes in any mathematical model there might be some things that might not be provable, so you have to do fit things as
yeah,
best you can
yeah.
to real.
Development of the model, right? So they didn't actually, did they not grab a data set? Yeah, it doesn't sound like they grabbed a data set on it.
Doesn't seem like they did. So, so models are quite interesting in terms of like analyzing the philosophy of like, okay, right. So, what are they assuming, and how do they think these things play into each other? How do you think these effects interact? So that's why
Yeah,
it's kind of interesting to think about infection models and like so we previously talked about paper where they look at mobile phone data and correlating that to different strains and that there's a lot of modeling where it's quite interesting like seeing how those interact. Just
yeah.
but that's
That
like the theory.
model plus the data that uses it. But yeah, this is just the pure model itself, not yeah, not the data to then see, oh, and this kind of matches up with some observations that we've seen.
Yeah. So I mean, it's I mean, again, this is the forefront because this is such a kind of difficult thing to study. So
Mhm, mhm, mhm. Yeah, yeah. I guess like it's kind of interesting. Like now that the way that I would think then about answering the question is now that there is a model, right, to try to describe this, right? What are the data that we could get? And then what are also like the what's possible, right? Because like
yeah,
there'll be something that's impossible to gather for this model.
mhm,
So then you have to kind of do further information to split out then what is the best substitute for that and arguing from the standpoint of here's what we're using the maths to show us. Yeah. Yeah, because you can make math show you anything.
yeah. I mean, that doesn't mean it's not useful. I think there's a lot of very interesting things that you can do about it. But I think like from our perspective, we need someone who's like au fait with that those kind of mathematics so they can see whether there's a hole in their assumptions or not.
For
Um,
sure. Sure. It's
so
not us. It's
yeah,
not
it's
us.
not us. We're not the right people for this. I don't think.
It's not us. Okay, we got 6-month consequences of COVID-19 in patients discharged from hospital, a cohort study.
Yeah.
I think this was a nice like seems like a nice big set of data, right? To analyze these long COVID
Yeah.
side, I guess I was calling them side effects, but they are symptoms, right? They're just
Yeah.
long-term symptoms.
It's a disease sequel. So you like the first disease, now you're going to get a sequel, which is just
No, we're in the age of sequels, even even
yeah.
for
I
this.
even even for diseases, it's a whole SARS is a whole franchise. We had SARS-CoV-1, we had MERS, which is kind of a spin-off, and now we've got SARS-CoV-2, which is like the Empire Strikes Back of SARS. Which means we don't have
Uh,
to worry about any good SARS disease from now on, unless they make a
yeah.
SARS Mandalorian TV show. There's not going to be any good SARS anymore.
But yeah, I think we said this a couple news things back. We haven't talked that much about the long term
Yeah,
um, right? I mean, that also includes like the clotting stuff we haven't really directly talked
Yeah,
about, or like autoantibodies. This is like more of a clinical study style, right? Where we're seeing like a big population of people and like what actually do you see out there, right? To try to get a sense of what we need to explain mechanistically.
yeah. I mean, it's interesting because they do like questionnaires where they ask people, they go out and ask people what they've been feeling. So find that like fatigue and muscle weakness were quite common symptoms, and sleep difficulties as well, which is something I've also
Mhm.
heard around from other studies. Just and also they brought people back in to do lung function tests and blood tests as well for antibodies, which I thought kind of I didn't look too deep into it, but that's like kind of an area where you want to look at for long COVID or for immune sequelae is one of the things that happens in diseases. So the
Absolutely.
disease I used to work on was strep pyogenes, so that had a few diseases that were sequels to it, like where it accidentally raises antibodies to the heart or to other areas. So it would
Hmm.
so you'd get all these autoimmune diseases that would come as a result of you having this infection.
Yeah. Yeah, I mean that's the
So
cost of having such a complicated immune system, right? Is that sometimes diseases will impact us a little bit later.
yeah,
Okay. What else we got here?
structure of SARS-CoV-2 ORF8, rapidly evolving immune evasion protein.
Ah,
So
yeah.
yeah, so this is a protein that was mutated in B.1.1.7, so there's a deletion in it.
Mm,
So this doesn't mention that at all, but it does talk a bit more about what this protein's function is. Um, so this one is kind of interesting because like original recipe SARS had a version of this protein, and a mutant appeared which had a mutation in this that made it less severe.
yes.
And so this paper dives into the actual structure of that protein to see how that relates to its function. So it's a very structural paper, lots of very interesting 3D images coming out about what this does and what it binds to, and then how it functions based on that.
Yeah. I mean, I'm always interested in all these non-structural proteins, right, floating
Yeah,
out in SARS because it's like it's the stuff that from the pathogenesis point of view, right? Like they're the effectors, right, that are interacting with our bodies and immune systems to create these effects that we get. So always very
yeah.
interested in that.
Whereas me, I'm just a hipster, and I'm looking at the things that people. Oh, you didn't hear about ORF8? I was heard about it 3 months ago. Kind of. Yo, you're going crazy with Spike. I'm on ORF8. No one's talking about
um,
it, and it does something, and I want to know what that is.
yeah, totally. We got structural basis for translational shutdown and immune evasion by the NSP protein of SARS-CoV-2. We actually had this in as a preprint before, and it got published, I think is why I'm putting it in now.
Yeah,
Um, and yeah, NSP1. We also talked about this briefly when they did like a big overview of like the SARS-CoV-2 proteins that bind RNAs in the body.
yeah,
Right, and they picked out this one, right, and they had this great story about how it's like shutting down translation of maybe they were suggesting interferon stimulated genes.
yeah,
uh, so here they're just zooming in on what the structural reasons may be for this particular protein to do that type of function.
yeah. So yeah, lots of like do cryo-EM, so they look at how the proteins interact in real terms, and then they look at see how that relates to like interferon. I mean, I'd be interested to see whether there's anything like specific they can say that this downregulates interferon specifically, because when you attack the ribosome, you attack basically every single protein. So of course
Everything.
interferon will go down. So
Yeah,
trying to
yeah,
so you can argue anything like oh yeah, it shuts down like pro-inflammatory proteins, it shuts down anti-inflammatory proteins,
yeah.
shuts down anything. So you can like pull out a lot of different stories. So I want to be looking for how they
Right.
kind of prove that this is a specific thing for immune evasion.
Absolutely, absolutely. Okay, and the last thing in our list, I think we have comprehensive in vivo secondary structure of SARS-CoV-2 genome reveals novel regulatory motifs and mechanisms.
Yeah, this is so the SARS-CoV-2 has this genome that's made of RNA, and when it goes and it's all tangled up in like kind of a
Hmm.
weird. It's got this secondary structure, so all the RNA is kind of knotted and it's in a weird shape. And this one kind of tries to map out that weird shape and figure out whether it has like any regulatory mechanisms. Like, does the shape of it affect what genes are being transcribed or and what aren't
Totally.
being transcribed, and also potentially like how can we obviously at the end of every single paper like this they go well is there a drug target, is there a drug target,
Yeah.
can we Even if there
Yeah,
isn't, this like, oh yeah, there could be. There's this chemical that we
there
produce
could.
it. Yeah, there could be. But
I mean, this is also dovetails right with the NSP1 story, right? Because
mm,
I'm pretty sure in that thing it was like it's a specific structure that they found that allows the SARS-CoV-2 RNAs to not be affected by the global shutdown
yeah,
that's enacted by NSP1. So right, like this is there, everything's linked. Biology is just like a real complicated linked mess of things. You'll always touch other topics as we talk about them, but like this is a great overview of those structures, because also some of those structures could have like enzymatic function, even like
yeah,
there's some crazy stuff RNA can do. It'd be interesting to see what they're doing.
yeah. So I mean, I picked this because it was just like something that I'd never thought about. I mean, so
I mean, I don't know how one would approach doing a screen, like just like I mean naively, like I'm sure if I spent maybe like an hour or two thinking about it, like I don't know how you would approach finding these structures.
yeah, that's in. So there's a
Like
lot
in
of
some kind of biased way where they like I mean they call it shape map, it sounds like, but what exactly is shape map?
yeah. I mean, that's interesting. It's curious. It's like something that I certainly wasn't taught when I was doing my microbiology degree. Something I never encountered when I was doing my PhD. So it's like kind
Exactly.
of an opportunity to learn a new thing, which is both
A new tech, a
yeah,
new crazy technique on how to untangle some of these functions. Yeah, and take a lens to biology, which I mean, that's why we love reading papers, right?
yeah,
Just like that lens on biology. Okay, so we got to choose a paper from this mix.
right, yeah,
What should we? Oh, and then the other thing, as we were talking, I was reminded that we do have that paper about inactivated virus. It was a structure. They used that a common inactivating agent to do a structural thing on the spike. I
right.
think that might be real-time conformational dynamics of SARS-CoV-2 spikes. Oh no, no, that's the fret one. That's the fret one.
But there's one that where it was like just real time looking at how Spike fuses. Is
Yeah, 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 yeah.
because
No,
I
but that's what I was thinking about when we were talking earlier about the inactivated viral vaccine.
hmm,
I thought that we had a paper. I must have hidden it somewhere. It's not actually in this top list. Um, where like they did the structure of the spike as it was inactivated. Here
right, okay. I
it is.
didn't.
Architecture of inactivated SARS-CoV-2 post-fusion spikes revealed by cryo-EM and cryo-ET. Louis et al. Uh, because they use, did they say what they use? I think they use that what they use when they make vaccines to inactivate uh this object.
Right,
So it's just like a structural.
let me pull it up quickly. It's taking a while. It is so odd how long PDFs take to put on my computer. It just
Oh. Yeah,
they're
well, yeah, downloading from the server. I think that we should tell this to people like that's the great thing about Zotero, right? Is that like we're putting these papers there, so if you join the group, you can download them for yourself and read them. It's a very convenient way to have a common group of papers to speak about.
see, it's still loading, but hopefully it'll come. Yeah, okay. Yeah, I've got it here.
Mhm.
Yeah, beta propiolactone. So yeah, that's the same thing uh, that they look at.
That they use to inactivate viruses for the vaccine.
Yeah,
So
that's
so
right.
what you're seeing is um yeah, you're seeing what that vaccine looks like really, right? Like this is an insight to structural biology of that particular type of vaccine, and then you can start to talk about maybe what it might. I guess it just gets recognized, right? It's probably inactive, right? And it just gets recognized. This is the shape of that vaccine.
Right. Yeah. Um. Okay. Yeah. This.
Really, I don't know if you want to do something more right? Because that's structural biology. We do a lot of
Yeah.
structure, basically because there's fun pictures to show.
So, because I'm kind of because the thing that people would like definitely like to see like I mean the Moderna vaccine is something that I'm sure people would want to watch, but it's kind of boring for us because we've done the same sort of thing at least 3 times in a row. Um, so it's very like samey for us unfortunately. But if you want to read it, you can read it. You can look at our old videos, and then get an idea of what you're looking for, and then dive in to it for
Yeah,
that kind
that's
of way.
definitely. Yeah, unless someone explicitly wants us to read it, I would say, yeah, it's good to see like how you read those types of vaccine papers, but then ultimately, right, like this one is another one that works. I agree. I don't want to necessarily read it unless there's like some burning desire out there.
Yeah, I mean, there's the of the B.1.1.7 papers. I think the one that is the most interesting would be the estimated transmissibility. Uh, so that's the one that was done by London School of Hygiene Tropical Medicine. So I can't.
Yeah.
So that's the one that has like a lot more modeling in it though. So, but they do pull in mobility data from sort of smartphone users. But in terms of like actual like what I'd be really interested in personally, is going to be like one of those like last few genome the genome paper of like secondary structure. So, but then
Hmm.
there's lots of other great papers here that we could look at.
Um, I mean, the transmissibility is interesting. Like, I also am actually I am interested in reading the transmissibility one a little bit, just because we did read. I I keep saying this, but like when we covered the D614 gene mutation, it was like so in post of it happening,
Yeah.
right? Like it was like, oh, it's already happened. This is like everywhere already. But here's one where it's like emerging. I'm interested to read it for that purpose, to like be at the edge of reading about this emerging mutation and just like maybe testing my own inferences about evolution, right, and my intuitions there,
Yeah.
like with this story.
Yeah.
So that's one reason I would want to read that. I mean, I guess it's nice that it dovetails with what people are interested in potentially.
Yeah. I think what other things are that we could because we do, do,
Yeah,
do.
I in vivo secondary structure, that's also fascinating to me. I always want to learn a new technique. As I said, I don't know that
Yeah.
uh, beforehand.
But I know the audience would be very much not interested in that because it's very much like a researchy sort of thing. Very much like how does the miracle of life work, and that's the
Yeah.
kind of like paper it is. And so I understand that people are like, okay, so tell me whether they're gonna be safe or not, and I'm going like, isn't
I
it
also
fascinating
have
how this
the
virus works?
olfactory transmucosal SARS-CoV-2 invasion as a port of central nervous system. That title looks wrong, but let me read it again.
So wait,
Yeah, as a port of central nervous system entry in individuals with COVID-19. Yeah, I was like, that can't be the whole title. As a port of central nervous system.
yeah,
That's the one where it's like we kind of previously talked about the neuropilin receptor, but
yeah,
they didn't really dive deep into where that could be, and this one dives a little bit deeper into that.
yeah.
Lots of
So it's got lots of like because it takes this is from autopsy samples of people, and
yeah.
they take a look at like where the SARS-CoV-2 is distributing, and also yeah. So this looks at like also microthrombosis. It does a lot of like things that we haven't necessarily talked about that as much. So
Mhm.
so yeah, I mean, so I think that either like this or the transmission of the new strain. I think I could be either or on these.
This or the transmission? Okay. I want to do transmission
Yep.
because between the two, I want to do transmission because, like, yeah, again, I want to test my I want to test my evolution intuitions.
Yeah. I kind of want to delve into like I want to test whether I'm able to understand how these models work because I feel like
Sure. Yeah, it is also like yeah, to really because like they're making assumptions in these models, right? And like how is that impacting the result that gets sput out?
yeah. So
Spat out.
yeah.
Okay, so that I agree. So 6 month consequences of COVID-19 in patients. Oh, sorry.
Sorry. Now
I'll just read.
estimated transmissibility and severity of novel SARS-CoV-2 variant of concern 2021-2/01 in England. Uh,
Yes.
so
Davies et al.
yeah, Davies et al. Is preliminary, not peer reviewed, so we're going to be trying to do our best to critique it. But obviously, this is going to be stuff that we don't necessarily know, so we might miss out stuff. But if you in the audience have read this paper and have one,
For this paper. And yeah, we're gonna cover.
yeah, we're gonna cover it. So this is great. So yeah, I mean, uh, let me bring us to the outro. Yeah, so join us next week for our deep dive week where we will cover this paper. Um,
Yeah, we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible that we didn't get everything right. So science is about thinking critically and asking the right questions. If you have any questions or corrections, please let us know in the comments.
yeah, I agree. And you can reach out to us over Twitter with the hashtag microTWJC. We both believe that peer review is a process which we can all participate in, and I hope you all have a good time listening to us ramble about microbiology today. I think if you think you have something to add or have found something unclear, please let us know because we want to bring that up on the show and help us communicate better about the science.
Yeah, it's been a pleasure chatting with you, Fuzz.
Same here, Danny. Tune in for more microbiology content next week, where we're going to talk about all things big that are small. Bye.